Today, we are diving deep into the really surprising and, frankly, kind of dangerous story of coliform bacteria. So, for more than 100 years, we've relied on this one biological detective to tell us if our water is safe to drink. But what if that detective, what if it went rogue and turned into one of the biggest public health threats we face today? Well, this isn't some sci-fi movie plot. It's the real story of coliform bacteria, a group that went from being a simple, helpful indicator to a whole family of deadly, drug-resistant superbugs. And this threat, it's not just some abstract idea. It is picking up speed fast. The very latest data out of Europe shows a staggering 60% jump in bloodstream infections from just one of these superbugs, Klebsiella pneumonia. And that's just since 2019. This is the front line of the antibiotic resistance crisis, happening right now. Okay, so here's our game plan. We're going to start with their origin story as the original poop detectives. Then we'll trace how they grew into such a troublesome family tree. We'll get into the nitty-gritty of how they build their superbug defenses, how doctors are fighting back with a modern playbook, and finally, what the future of this whole battle looks like. So how in the world did this all get started? Well, it began with a really simple but totally brilliant idea. Instead of trying to find a few rare, dangerous germs in, a literal ton of water, why not look for their buddies who are always hanging around? Let's go back in time. It's 1855, and John Snow just proved that cholera is spreading through the water. All of a sudden, everyone realized we desperately need a way to know if water is safe. But trying to find the actual cholera bacteria was like looking for a needle in a haystack. So in the 1880s, scientists had a thought. Let's look for something way more common, coliforms, these bacteria that are in massive numbers in all animal poop. By the time Theodore Estrich identified what we now know as E. coli in 1885, the strategy was basically set. If you find coliforms, it means there's fecal contamination, which means there's a really high risk of finding other, more dangerous stuff too. It was an elegant, perfect workaround. But here's the twist. That simple coliform group? It's not just one or two harmless bacteria. Oh no! Thanks to modern genetics, we now know it's a huge, sprawling family tree with 19 distinct branches, and a lot of its members are anything but harmless. Let's run down the family's most wanted list. Of course, you've got E. coli, the most famous one, but it's also the #1 cause of UTIs and deadly sepsis. Then there's Klebsiella pneumoniae, a huge driver of pneumonia, and basically a factory for creating and sharing antibiotic resistance. You've also got hospital-acquired threats like Enterobacter and the especially scary Citrobacter cocceri, which can cause brain abscesses in newborns. It's terrifying. You see, these bacteria kind of lead a double life. They're totally normal in our gut, but if they get into the wrong place, they can become absolute killers. Now for the really fascinating part. How do these bacteria actually defend themselves? They haven't just stumbled upon resistance, they have engineered some incredibly clever defense systems. So let's take a look inside their arsenal. Okay, first up is what I like to call the booby trap defense. The technical name is AMP-C induction. It's basically a genetic tripwire. Certain bacteria, like Enterobacter and Citrobacter, have this super powerful resistance gene called AMP-C, but it's usually switched off. The moment they get attacked by specific antibiotics, bam, they flip a switch, unleash this enzyme, and it just obliterates the drug. And here's exactly how it works. It's brilliant. First, an antibiotic like ceftriaxone comes in and starts damaging the bacteria's cell wall. Step 2, all this debris from the broken wall starts piling up inside the cell. Then, step 3, a little sensor protein detects all that junk and acts like a tripwire, flipping the genetic switch for that AMP-C gene. And finally, the counterattack. The bacteria unleashes a flood of the AMP-C enzyme, which totally neutralizes the very antibiotic that's trying to kill it. It literally turns our own weapon against us. This all leads us to the ultimate threat, Carbapenem-resistant enterobacteriales, or CRE for short. You might have heard these called nightmare bacteria, and for good reason. They're resistant to carbapenems, which are some of our most powerful last line of defense antibiotics. Their superpower comes from these destructive enzymes called carbapenemases. Now, these devastating enzymes come in 3 main flavors. You've got your Class A enzymes. These are the most common, like the infamous KPC enzyme. Then you have Class B, which I call the metal eaters. They literally use zinc atoms to chew up our antibiotics. And finally, Class D, the sneaky OXA-type enzymes, which are super tricky for labs to even find. Knowing which one you're up against is absolutely the key to winning the battle. The good news is we have developed some countermeasures. For those common class A enzymes like KPC, we now have drugs like avibactam. You can think of it like a shield. It just gets in there and blocks the KPC enzyme, basically disarming the bacteria so our main antibiotic can do its job. But those class B metal eaters, they're a whole other level of tough. Our new inhibitors don't work on them, so this requires a much more clever strategy. Doctors will use a two-drug combination. One drug, astrianum, can kill the bacteria, but it's vulnerable. So the second drug, avibactam, acts as its bodyguard. It goes in and knocks out all the other defensive enzymes, so astrianum can get through and land the final blow. It's an amazing strategy. So, how do doctors actually put all this knowledge in the practice on the front lines? Well, they now have a very precise playbook based on the latest 2024 guidelines for choosing exactly the right weapon for the right bug. And this is that playbook. It's a really clear if-then kind of strategy. If the lab finds a KPC enzyme, you use the drug combo where avibactam can block it directly. If it's an MBL, you use that clever bodyguard strategy with avibactam plus astrianum. And if it's one of those sneaky OXA48s, it turns out avibactam works there too. This is a huge shift away from just broad-spectrum guessing to precision-targeted warfare against these superbugs. So what's next? I mean, this arms race is definitely not over. Let's take a look at the next generation of weapons currently in development and talk about the bigger picture of where this fight is really taking place. Coming down the pipeline very soon are even smarter drugs. One is called taniborbactam, and it can be a total game changer because it can block both the common KPC enzymes and those difficult MBL metal eaters, all in one molecule. Then there's zidibactam, which is even more clever. It's called a beta-lactam enhancer. Not only does it block the resistance enzymes, but it also directly helps the antibiotic kill the bacteria. That's a true dual-action attack. But here's the reality. New drugs alone are not going to solve this problem, because the battlefield isn't just in our hospitals. A recent genome analysis confirmed that the exact same resistance genes are being found in bacteria from both humans and pigs. This is clear proof that our food chain is acting like a critical highway for spreading antimicrobial resistance. It all comes back to this one health idea, that the health of humans, animals, and our environment are all connected. We have to tackle all three to win this war. So we've got on this journey from a simple poop detector to a full-blown global superbug crisis. It really leaves us with a critical question for our modern world. What happens when the very tools we design to protect ourselves evolve into the very threats we fear the most?